This is TWIM, This Week in Microbiology, the podcast that explores unseen life on Earth. This very special episode of TWIM was recorded on Friday, June 14th, live from the exhibit floor of Atlanta's own Georgia World Congress Center, where our audience is experiencing the annual gathering of microbiologists at this year's ASM Microbe Meeting. I'm Michael Schmidt, and joining us for our live session from Tacoma, Washington, where he's on the faculty of the University of Puget Sound and was the 2018 recipient of the ASM Karski Award for Excellence in Undergraduate Education, is the host of Microbial Matters, the seemingly ever-adjacent TWIM, Mark Martin. Welcome, Mark.
Thank you very much. Did all of you get your if you didn't get a 3D printed tardigrade from me or a pen, I will be around later.
So, Mark, folks may not know the significance of your Karski Award, which was established in 1963, where its first recipient in 1969 was Dr. Sid Rittenberg from UCLA, a legend in our discipline. And it's an annual honor bestowed in recognition for excellence in undergraduate education and is made in memory of Ted Karski, who I had the pleasure of meeting once, who founded the Baltimore Biological Laboratory that many of us recall and have used the products from BBL. So again, Mark, welcome. And for folks who have not yet discovered Microbial Matters, one listen and you will all understand why Mark's a Karski laureate. So thank you, Mark, for stepping in. Our other host of TWIM, the ever-famous Michelle Swanson from the University of Michigan and Petra Levin from Washington University in St. Louis, are busy. And so they can't join us today and send their regrets. And of course, our podfather of all the Twix podcasts, Dr. Vincent Racaniello, is just back from Europe, and he didn't know if his plane was going to get here on time, so he too sends his regrets and tapped me to host. But fear not, we're going to give you some science today, and we're going to have some especially cool science. So let us please welcome Dr. Arturo Casadevall, who is, and I got to get this right, so I have to use my cheat sheet, the Bloomberg Distinguished Professor and Alfred and Jill Summer Professor and Chair of the Department of Molecular Microbiology and Immunology from the School of Medicine at the Johns Hopkins University in Baltimore. Dr. Casadevall has authored over 1,000 publications. I'd hate to print your CV. And was recently inducted into the National Academy of Sciences and has gone entrepreneurial on us by dabbling in the space of inventing new ways to cool our buildings, albeit using fungi, and offers courses with some of the most provocative titles I have ever seen. Wouldn't you like to take a course on how do you know, the theory and history and practice of science, the anatomy of scientific era, and metascience in research and practice? So welcome, Arturo. We thank you for joining us today. And as the title of our session, or this week's edition of TWIM, is, Could Fungal Pathogens Outsmart Us? And this is so the folks at HBO don't come after us for talking about The Last of Us. So Arturo, welcome.
Thank you so much for having me, and it's so much fun being here. I thought that for Marco Martin, I thought I would read you what I wrote on his...
Please do.
On his, he handed me the book, so I wrote, for Mark, one of the greatest educators of our time, education ensures the future.
Hear, hear. So I've known Arturo for many years, and in the early days of SARS-CoV-2, before it was declared a pandemic, so this was late February, I was chair of the Council of Microbial Sciences, the think tank of ASM, and we were tasked with developing our society's response to what we all knew was going to ultimately become the pandemic it became. So I reached out to Arturo and I tapped him to pick his brain about this emerging threat pathogen. And he was, of course, very busy at the time. Those of you who know his CV know he's been heavily involved in the early days of the pandemic and continues to work on that particular topic. And I reached out to him to get one of his folks to talk at our symposium, which was in early March before the WHO declared the pandemic, specifically about the opportunities that convalescent sera could offer.
Right.
And at the time, we were beginning to lose thousands of people per day to this virus. So we have a lot to talk about. And I'll start with one of the softball questions, simply to help our audience appreciate the significance of things. Human and fungi share 50% of our nucleic acid, 50%. So how can we shift this dynamic balance so these wee beasties don't get the upper hand and outsmart us?
Well, they already have the upper hand. They are the most successful kingdom. The fact that they are so similar to us is fascinating, because when you run into a mushroom in the forest, you don't see your resemblance. I certainly don't. Maybe. But the fact of the matter is that it's very serious, because being so similar means that there are very few differences to target in drug development. And consequently, it is hard to make antifungal drugs, and we have very few.
And, I think, this is one, I teach medical students and dental students for my day job. And one of the questions that I always get asked is, why do we have 200 different species of fungi on our feet?
They certainly like our feet. But I think the message there is that there are 6+ million fungal species, and that they occupy any niche possible, and that we often don't think about them, but that they're in your skin, they're in your mouth, they're in your gut, they're everywhere, just like bacteria are. And I think people who study the microbiome needs to pay particular attention to that, because when you change a variable that affects the bacteria, you're also changing the fungi, you're changing the amoeba, you're changing a lot of other things.
It's all about...
Right. It's all connected.
Yeah, and it's a fitness function. And I think there's not enough attention paid in the manuscripts that are coming out on the microbiome. We're not paying attention to the viruses in the community. That's my plug for Vincent. As well as the fungi that are interacting with these creatures. And one of the things that, again, terrorizes me at night, things that keep Mike up at night, is fungi thrive on disasters. I happen to live in Hurricane Alley, and so hurricanes start June 1st and last till after Thanksgiving. And fungi thrive on disasters. And one of the most fascinating stories that I didn't know that I read when I read Arturo's book is that the fungal pathogen that started in the Pacific Northwest, Cryptococcus gattii, is a consequence of a tsunami. Now, why don't you tell that story of how Cryptococcus gattii effectively got to the Pacific Northwest, established itself, and the rest, as they say, is history.
So I want to begin by saying that it is a theory. It's like a lot of these things is putting together a lot of ideas. But the origin of this organism is northern Brazil. And the idea is that when the Panama Canal was first opened, it was carried in its ballast, and it was deposited in the area of the Pacific Northwest, where it sat for 60 years until the 1964 earthquake created a tidal wave and then deposited on land. But that opens, so that, it's the kind of thing that you can connect the dots. Do we know that that's what actually happened? We don't, but it's quite possible if you look at all the pieces of it. But it also gives you a mechanism to think about how organisms that can get washed into oceans can find their way back into land and perhaps alter the ecology and the diseases in certain places.
Mark?
I think this is a fascinating idea. And what I tell people is that if you look at the Amazon rainforest and you pull out one member of it and you put it somewhere else, it perhaps won't grow well. But on the other hand, I have houseplants in my office, and I've seen identical houseplants in Hawaii that are 10 times its size. So it all comes down to context, context, context. And what we've got is a situation where we're walking around as a series of ecosystems, and we don't think that way. And just because we have ecosystems, we believe that they're stable, and that's not how ecology really works. And so you can have these disruptors, for lack of a better term, when they get, and I'm not going to say a foothold, a rhizohold. What would I say if I wanted to be fungal?
A fungal hold.
Yes.
Fungal hold.
And then they can start to develop from that, and as they get advantages, those things spread. And it's something to keep in mind that we're just a single star in a constellation of microbial matter, and we got to remember that, I think.
I mean, if you can put it into one sentence, the fungi do well whenever the earth does badly. In fact, there is fossil evidence. The greatest extinction in the world is the Permian extinction. And there is a mountain in South Africa where there is 1 meter of fungal spores. So as you kill off vegetable and animal matter and all that, the fungi thrive.
1 meter of spores. That gives a whole new concept to your shower stall. Think about that the next time you're getting rid of the red scourge. So, how many of you had a banana for breakfast this morning? How many of you know that the banana is endangered?
Quite a few.
So, the banana story is another remarkable how the fungi are really outsmarting we who think we're smarter than everyone.
Bananas, wheat, rice, or main staples are all threatened by fungal diseases, and it's a continuing battle, and it's often a losing battle because they're getting resistant to the fungicides. And many of our crops don't have a lot of genetic diversity anymore, which makes them very susceptible. And if we were to lose 2 of the major crops, or 3 at the same time, imagine losing rice, imagine losing wheat, humanity will face starvation because there is no way to replace those calories.
That is indeed the case. And if you think about it, maybe we should be moving away from these inbred lines that we're effectively promulgating because they produce so much food. And in last night's opening session, for those of you listening that didn't hear it, we heard that by the year 2075, the planet's going to have about 10 million souls on it that we're going to have to feed. And that's going to compete for area, it's going to compete for food, and we don't have any easy fixes at this point in time.
I think that our best bet is the continuing biological revolution. That is to engineer a lot of these crops to make them resistant. It will always be a battle. There will not be a day in which you could declare victory, but you could stay ahead of it. So I do think that what people in this meeting do, which is to do research, to figure out mechanisms of pathogenesis, mechanisms of physiology, can be the solution for this century.
It's important to remember, we were talking about bananas a moment ago, that most bananas are triploid. They don't reproduce sexually. They're done by cuttings, which means they're identical to one another. So if they're sensitive to a particular disease, and fungal diseases are part of that, then they're all sensitive to it. And why did this happen? The idea was these are highly productive cultivars, and we want to keep them, but we need to be nimble. We need to be moving ahead of it, and we're not good thinking ahead. We're good at putting out fires as they happen, I think, but we don't think ahead, and we really should. And this is a really good example.
Mark, humanity was not selected for long-term planning.
That's true.
People say, I don't know what's going to happen tomorrow. How can I worry about a month from now? But we need to really, really rethink that, because just whereas our nature is not inclined to long-term planning, our survival is.
I agree.
Yeah, it's absolutely fascinating. Staying on the theme of antifungals and our crops, another fascinating piece that you speak to in your book is about in hospitals where the patients were acquiring fungal diseases that were resistant to our first-line antifungals, and they just so happened to be in the Netherlands, which was very famous for its massive tulip fields, and they were protecting the tulip fields with antifungal pesticides to effectively ensure the success of the flower fields. And the hospital so happened to be near the tulip field.
So here's the fascinating thing. They were using azoles in agriculture, but the thing that was affecting the patients was not the target.
Correct.
In other words, it was sitting in the soil. So when people spread these things, you're selecting for resistance in organisms that is not necessarily the ones you're targeting. And that's something that we often don't think about.
Yeah, and that brings me to my next question, is how did you get to where you've become this expert on convalescent sera, a Bloomberg professor of public health, a physician, and most importantly, I think your primary role is scientist and science educator?
Well, you're very kind. You know what? I'm interested in a lot of things. And if I can give people in training one advice is to take 5 minutes each day to read outside your field. It doesn't matter what it is. But if you do that for 1 year, you have 365 new factoids. And eventually you begin making connections. And this will affect how you think, and it will affect, how you put things together. And that's what I try to do. I try to read outside my field every single day.
I think that's great advice for all you young folks in the room, is read outside your field. I've certainly followed that practice. Mark, have you?
What I was going to say is that's one excellent thing. This is a plug, actually, for Arturo's book, let's be clear, which is a wonderful book in lots of ways, and I'm happy to talk about it. He embarrassed me. I could do that right back. But what's important to say is the section where he talks about his advice to going outside the field is something that's not rewarded. We're always told we need to really specialize, and I understand where that's coming from. But you see better with more perspective than less. And we get too centralized, we get too narrow. And coming in from outside, I can't tell you the number of times, even in my own little lab, where a student who hasn't had genetics, they'll ask a question which completely changes the way I looked at things. And so I really recommend that idea that you just heard Arturo state. Read outside your field a little bit every day. And by the way, that doesn't mean Google.
I think most of us went into science because we were curious. And then when you get into a scientific training, they tell you focus, focus, focus. Focus means getting narrower and narrower and narrower. And that's important because to do major work, you need to go deep. So there is no question you need to focus and you need to go deep. What I argue with is that you should not lose what you came in with, which was your curiosity, and also to look outside of it. And it doesn't take that long to do. And once you get into it, it's like going to the gym, You want to learn more, and you want to learn more. And I would just say that it leads to a fuller life.
No, it does indeed. And, going back to your involvement with the SARS-CoV-2 pandemic, give us an update on what's going on in the world of convalescent sera. Is it still
Yes.
as useful as it was 100 years ago?
So in January of 2020, once I heard that asymptomatic transmission was occurring, I knew this was not containable. And I also heard that people were talking about antivirals, vaccines, and monoclonal antibodies. But nobody was talking about an old therapy that was known to be effective for more than 100 years, and that was convalescent plasma. So the word is, how do you get the word out? I could have put it in MBio, but I don't think that would have connected. So I wrote an op-ed and began to send it to newspapers. New York Times didn't want it, Bloomberg News didn't want it, Washington Post didn't want it, but everything in life is luck. I sent it to the Wall Street Journal on the day that the market tanked.
Ooh.
And here was a bit of good news. So they printed it. So why is that important? Because now I had a link that was really well written with 800 words. And I could send that around to everyone. So we were able to mobilize, and within a few weeks, as a result of thousands of individuals who mobilized, this country went from 0 units of convalescent plasma in March 1st to using it in thousands of people by May. And after the first year, 600,000 people were treated. And we have evidence that it saved 10,000 of lives. And then it basically is no longer needed. Why is no longer needed? Because everybody has antibody as a result of either having infection or having the vaccine, except for one group, the immunosuppressed. So convalescent plasma remains used today in the immunosuppressed population because it's giving antibody. So it continues to be used in a narrow group of individuals who don't have antibody and need it.
And I think one of the most important lessons learned from the convalescent plasma story is one that Tip O'Neill taught us. Like politics, it's all local.
Right, sir.
And your convalescent plasma has to be local.
That's right. So that's one of the things that we learned. So early on, for example, many clinical trials try to centralize the supply of plasma, but that meant shipping it across the country. And when the analysis was done, this is a very variable virus. Every city has their own, you use the word quasi-species. So it turns out that the efficacy of plasma dropped with distance. So if you went over 150 miles, it really dropped. And
Just
that was
like
one
politics.
of the earlier indications that we were dealing with something very variable.
And I think that's what we're learning about this virus is we all know it's a messenger RNA virus, and one of the things we learned in virology is mRNA viruses lack what? Proofreading. So it's not unexpected that they would change their spots. And selection is a powerful tool. And it's live or die in terms of reproduction.
I think you need to remember that the hand of Darwin is on all that lives. And we forget this, that we tend to be focused on things that don't change and things by themselves. And that's not the way it really works. It might be the way that you start to study something to think that way, but we know already from HIV how you can get almost individual fingerprints of versions of HIV. In fact, some would say to individual patients. So this shouldn't be a surprise, but it was, wasn't it?
Right. I mean, Mark, one of the things you brought up, Darwin, I think about Darwin a lot because for him to put the origin of the species together, he had to look at a lot of very different things. If he had focused only on the pigeons, if he had focused only on the birds, he wouldn't have put it all together. But I posed to you a question, where would Darwin publish today? He didn't have a mechanism.
That's right.
And they would have probably told him it's too diffuse.
No, you're absolutely right. And it's kind of frightening.
It's a problem with our times that thought is often not valued. Thought is really, really important, and we need to have more thought in biology and microbiology.
Well, we all remember the next battle hypothesis that's now fact, and that was the chemoosmotic hypothesis, the great Peter Mitchell.
That was so much fun for me because I was taking biochemistry from Paul Boyer at the time the Nobel Prize was announced for Peter Mitchell. He was not dealing with it well. And he said, and I quote, "If they gave a Nobel Prize for good guessing, Mitchell should have gotten it." But the fact of the matter is that the reason that Paul thought that is there wasn't a good stoichiometry. And he was so steeped in chemical thought that he didn't consider in a living system there are variable costs to move things across membranes, reducing equivalence or ATP proper. So it isn't possible to have an exact stoichiometry. So this is, once again, I think, underscoring the fact that we need lots of different ideas that people work with and come together, and not rejecting one another's ideas, but learning from them.
Yeah, in 2010, writing in MBio, which Dr. Casadevall is editor-in-chief still.
One more year.
One more year. You predicted the rise of the fungal threat, like Candida aureus, would be coming.
Right.
And so it did. Charde and colleagues writing in 2021 in MBio were able to isolate a multi-drug susceptible as well as a multi-drug resistant form of this fungus from the salt marshes of all places on a beach on an Indian island in the Bay of Bengal.
Right. The Andaman Islands.
Yeah.
So the idea is very simple. It turns out that the reason most people don't worry about fungal diseases is because you have tremendous resistance to them. And that's because your temperature keeps out most of the fungal species. But what would happen if the fungi adapt? And they're going to have to adapt with global warming. So the idea is very simple. There is a lot of fungi out there with a lot of pathogenic potential, but they're currently not a problem for you because your temperatures keeps them out. But if they jump over it, then we're going to see new fungal species, new fungal diseases. And this may be the canary in the coal mine, the first one.
So that brings up the next question about climate change and the fact that the planet's warming by significant degrees per year. We're seeing the melting of the polar ice caps. We're seeing the melting of permafrost. What should we be wondering about, and how can people who don't plan?
Well, the first thing I want to say is that we are the first humans who are witnessing climate change. That's never happened before. In our lifetime, things are changing. Usually it will happen over centuries, so you wouldn't see it. So in a way, it's remarkable what we're seeing. It's really bad, too. It's going to change everything. The question is how to prepare for this. Well, I think that no one thing is going to make a difference. I mean, we're going to have to basically take on the whole thing, from taking the carbon out of the atmosphere to preparing for new organisms that we haven't seen before in the permafrost or in adaptation. But if you ask me what we need to do, is we need to sample the natural world. We don't even know what's out there. We don't know what's out there because that's not the kind of science that gets funded. Try to write a grant to say you want to sample some of the soils or something like that. It's not going to go anywhere.
The fishing expedition.
Exactly. And yet we need to know what's out there.
So, Mark, you have the fortunate experience of interacting with undergraduates that come in all enthusiastic. So how are they receiving this information about the fundamentals of climate change and the microbial involvement and what we as an informed race can do about these problems?
Well, I think the first thing is that most students arrive to learn microbiology from me with some ideas they had heard of earlier. If they'd heard about archaea, for example, they've heard they're all extremophiles, which is not true. They'll hear that bacteria don't have organelles, and they do. There are all these kinds of things that they were taught before. What is the Mark Twain quote that it's not what you don't know that gets you into trouble, it's what you think you know and you don't know that gets you into trouble. And I think that's true. So when I teach my students, one of the things that's very important is for me to teach them, you've heard me joke about it, OMG for overwhelming microbial greatness, and WTM for what the microbe. These are things that I bring up in my classes, something they'd never heard before, something that really blows their mind, so to speak. That gives away my age a little bit there, but the point is things that they hadn't heard. Because most of the time, in most of the fields of STEM, things are very oriented in a hierarchical fashion. It's like when I'm asked, do you teach the structure of peptidoglycan? And I love to say, well, which one did you want me to teach? Because there are lots of different ones. So what I'm trying to say is one thing that I've taught them is how incredibly versatile, mobile, and flexible the genomes of microbes really are. And then they think, so is that colony on a plate truly homogeneous? And I have to break their hearts and say, why, no, it isn't. Well, so how do we find out anything? I said, that's the question for you. What do you think we should do for it? I'm easily able, when I teach, to have them find variants that swim faster or slower, and then we ask what genes have changed. And I said, you're now an agent of natural selection. Now think about the fact that that's going on all the time when you aren't looking. And that's the world. And so the key, and we've talked about this in terms of resistances, is that the antibiotic crisis that we deal with is wholly because we are staying static with those things, and the hand of Darwin is on the microbes. So we have to find a way to kind of use that level of variability back, which is why phage therapy has real promise. We need something very similar to deal with this fungal crisis. I don't know if people have tried in vitro evolution of fungi to see if they get better at higher temperatures.
So people have done that, and the scary thing is that you can get them to grow a much higher temperature, and when you sequence them, you need very few changes. It's not like you need to mutate the whole thing.
So that's bad news.
It's remarkable how rapidly these things can adapt.
Yeah.
You know, one thing is I urge you to think about is it's not the average temperature of the Earth.
Yeah.
It's the number of really hot days.
Mm-hmm.
Every hot day is a selection event.
That's right.
And if you survive and your genes can make it to the next generation, you have that information.
You know, the microbiologist Bill Metcalf, we were talking about a phenotypic assay many years ago using McConkey plates and sugars. And I said, well, now I know I have X. And Bill said to me, you know what? The microbes aren't in business to do this phenotypic assay. They have their own agenda, and it's not yours. And that's quite true. They were, first evolved, last extinct. That's the motto I came up with many years ago about the microbial world. They're better at it because they've been at it longer.
Billions and billions of years, to paraphrase Carl Sagan, I think. So the word that everyone is using around my university is AI, artificial intelligence. So how can we, as a group of concerned scientists working in the microbial disciplines, begin to use AI to our advantage so that we can outsmart our fungal friends?
Well, I don't know if we can outsmart them, but certainly AI can help you. And I think AI is one of these greatest things that have happened in our lifetime. And we are just learning. We're at the beginning of a revolution on using it. But I think what it does for you is it often generates lists, it generates solutions. Now, they may be wrong, but occasional one is one that you haven't thought about. And that is very important and very worthwhile.
Well, when I think about AI, I say to myself, what they have done is taken your advice. The AI bot has read everything it could get its hands on. And it then began to effectively associate and make comparisons and formulate hypotheses looking for data that supported the next thing.
That's right.
That's what AI is all about. And that's what we've all been doing as we do science.
That's right. And right now it's very primitive. You know, a lot of the things that it says, and you can tell are not correct. But this is only a couple of years.
It is.
I mean, and this is all going to get better. So I think that I like to think of AI as something that's going to help humanity get through some of these greatest crises.
AI is also going to be in the business of being good for AI. I think it's going to be trying to evolve itself, don't you think? I read too much science fiction, friends.
So I guess, looking at the elephant on the TV, that made it to HBO Max, and I believe that your phone was ringing off the hook when the series kicked off, and we don't know what season 2 is going to bring us. But how based on fact is The Last of Us?
So when The Last of Us first came out, I found out about it because I began to get emails. I said, the people asking me, were you the consultant for that? I said, what are you talking about? So I had to Google it, and I found it, and I take a look at it. I said, oh my God. It's based on this idea of temperature adaptation to bring you a new problem. Now, what everybody wanted to know, and I had my 15 minutes of fame with reporters talking to me, was, is this possible? And my answer was always the same: unlikely, but not impossible. And what did I say? What do you mean? I said, well, it's unlikely. We don't have any precedent of a fungus taking over somebody's brain and making a zombie out of you. But then again, fungi make LSD. And if fungi can give you a hallucination, well, who knows what it could do to you?
I used to teach a writing course for first-year students about symbiosis and parasitism, and yes, I'm aware parasitism is a form of symbiosis, but most people in my university were not. So that's why I called them both. People were in love with things like cordyceps, which is what The Last of Us is essentially based on. But there are other types of very strange things. There are viruses that infect caterpillars. Now listen carefully. When infected, they do something different than they normally do, called summiting. They crawl to the top of a leaf, and then they basically dissolve, sprinkling virus underneath. And I'll let the students think about that. And I said, why do people climb mountains? And I let it sit. How would we know if we weren't being manipulated? I don't mean to be all paranoid about it, but how do we know? What does the insect think when it crawls to the top? And then the conidia come out of its brainstem, right? Also, David Hughes's work on this is well worth your time. You'll find out that the fungus is actually an internal marionette. It's actually yanking the different body parts to make them move. It's fascinating.
I think you make a very good point. How could you know if you are manipulated? How do you know if you are manipulated to open the refrigerator door late at night?
You want that ice cream.
How do you know you're not manipulated to love microbiology?
Right. How do you know? I mean, it's actually a very profound question. And try to think how you would answer it if somebody gave it to you as a scientific problem.
So I guess one of the last questions I'm going to ask before we take questions from our audience is, we learned many lessons from the SARS-CoV-2 pandemic. Are we going to pay attention?
Humanity often forgets things, and we want to forget because it was not a good experience.
No.
But I also like to think that we retain information and we can use it. I like to think that we will learn. We have learned a lot, and perhaps if we face it again, we will do better the next time. But I'm an optimist.
Scientists have to be optimists. We've all thrown our grants over the transom and lit the candle at the cathedral and hoped. So, questions from the audience, please. We have a question down at the front. Ray's got a microphone. He'll bring it to you, so you'll be immortalized on the TWIM podcast.
So with being an optimist and scientist, is there any benefits that can come from increasing global temperatures?
You know something? The answer is going to be is local. It depends where you are. Some places will get better. I mean, and I pose to you a question. Imagine that humanity figures out how to take the carbon out of the atmosphere. Imagine a conference in the year 2090 to try to come up with a global treaty as to where you set it at. Because if you lower it too much, you're going to some areas that would then be warm will freeze again. So I would argue that that everything is a trade-off. You know, at the end of the Cretaceous, on that day that the dinosaurs went out, there were no ice caps. Sea levels were much higher than they are today. Life finds a way. But I think that there will be definitely trade-offs. But right now, global warming is bad for most of the things because we're not adapted to it.
I was wondering, in the fishing expedition context, how do we even go about maybe starting those things? Like, how do we, make them worth our while or show that they're worth our while? And how do we even take on such an insurmountable task? Like, where do we even start?
How do you start? That's fundamentally the question. You start with an idea, I think, and you refine your idea, share it with your friends that you trust to give you good criticism. And as it is criticism, you have to have a thick skin. But
Right.
Ideas are the bread and butter of science.
And then you do.
And then you do.
And then you do, because I think all of us know the experience that you think you know something, and then you begin doing it. And then you realize that the doing it teaches you a lot.
Don't let people tell you something doesn't work. If it's something that you can try, I promise you this. If your advisor tells you, oh, that's a silly idea, you should never do it. But if you hadn't asked the question, if you tried and it didn't work, so what? But I promise you, if you tried and it worked, you'll be a genius. And just because someone has a bunch of titles or is fancy doesn't mean they're any smarter than you are. Trust me on that.
So, I'd like to thank our guest here today, Dr. Arturo Casadevall, for joining us for this free-for-all. Thank you very much, Arturo. We look forward to reading more.
Well, thank you for having me, and thank you for being here and sharing this time with us.
And of course, our ever TWIM adjacent, Mark Martin. Thank you.
Thanks, folks.
We'd love to get your questions and comments on this episode and all our episodes of TWIM. So if you have them, drop a line to twim@microbe.tv, and we'll be happy to read your question on air and tackle it at a later date. I'd like to thank ASM for their support of TWIM and Ronald Jenkins for the music that you routinely hear, and for all the behind-the-scenes magic that takes place, especially from Karen at the Microbe TV Incubator, who keeps us on the straight and narrow on the days we record, and of course, the director, editor, sound engineer, and master of all our episodes sounding so good. It's Ray Ortega that you see here. So with that, we thank you for attending this very special session of TWIM at ASM Microbe 2024, and we'll see you next time on TWIM.